Hello students, so today we are going to discuss the next portion of sexual reproduction in flowering plants. Now we already discussed in the last two lectures about the anatomy of the male reproductive system in case of the plants and the female anatomy of the flowers in case of the flowering plants. After that, we have also seen that what do these gametes looks like, or what is the structure of this pollen grain, what is the structure of this ovule, and few more things. Now what we are going to study how this particular pollen grain gets transferred from the male part or the androecium of the flower to the gynoecium of the flower, right? So basically we are going to study another part of the pre-fertilization event, and that is what? That is the gamete transfer, right? So in case of the gamete transfer, I have said in the first chapter, if you go to the previous videos of chapter 1, as I have mentioned earlier, also I'll mention once again that if you haven't gone through those videos, I'll recommend you to go through those videos first and then come to this one, because each and every one of them are linked with each other, right? So we have already seen that this gamete transfer in case of the plants are called pollination, whereas in case of animals it is called copulation. So here we are discussing about the transfer of pollen grains or the transfer of gametes in case of the plants, which is also called pollination. So what is pollination? Pollination is basically, it is the transfer of pollen grains from the anther to stigma. It might sound new to all of you, but it's very simple and very easy. It is a process of what? Of transfer. Of transfer of what? Transfer of pollen grains. From where? From the anther. Now anther is found where? Anther is found on the androecium, right? So it is transferred from anther to stigma. Stigma is found where? It is a part of the gynoecium. So basically the transfer of the pollen grain from androecium to gynoecium is called what? Pollination. Clear? Now this pollination are of three different types. There are three different types of pollination. What are the three different types of pollination? The three different types of pollination are autogamy, geitonogamy, and xenogamy. Let us discuss each one of them very simply. If you see the diagram A, if you see this particular diagram, that is diagram A, in this diagram, if you see this diagram, let me mark this with this color. So if you see this diagram A, you can see that the transfer, this red arrow indicates the transfer of the pollen grain. So you can see that the transfer of the pollen grains is occurring from anther to stigma of the same flower. So it is basically what? Autogamy is what? Autogamy is the transfer of what? Transfer of pollen grain from anther to stigma of the same flower. Here you can see that the transfer is occurring into the same flower. Such kind of pollination is called what? Autogamy. When the transfer of pollen grain is occurring from the anther to the stigma of the same flower. It is called what? It is called autogamy. Right? Now if you see on the second diagram, if you see the second diagram, that is this one, in this case you have two flowers. One is a flower A and the other one is flower B of the same plant. Very important thing, flower A and flower B belongs to the same plant. As you can see here in the diagram itself, that we have a single stem, and on this single stem we have two different flowers. One is named as A, the other one is named as B. If the transfer of the pollen grain occurs from the anther to the stigma of two different flowers but of the same plant, that is called what? That is called geitonogamy. Make this thing very clear to all of you. Autogamy is what? Transfer of the pollen grain from anther to the stigma of the same flower, whereas geitonogamy is the transfer of pollen grain from anther to the stigma of different flower but of the same plant. Clear? Final one is what? The final one is xenogamy. The final one is xenogamy. As you can see here also, xenogamy. Xenogamy is what? Here you can see we have two different plants, plant A or plant 1 and plant 2. Plant 1 contains this flower that is named as A, and plant 2 contains a flower that is numbered as B. Right? So if the transfer of pollen grain occurs from the anther of one flower to stigma of another flower of a different plant, very simply, very simple thing, but very critical thing. What is xenogamy? Transfer of pollen grain from anther of a flower of a plant to stigma of a flower of a different plant, that is called what? Xenogamy. So xenogamy is what? Transfer of pollen grain from one flower to another flower of a different plant. Just like geitonogamy was transfer of pollen grain from anther to stigma of different flower but of the same plant, here it is different. Here it is how different? It is getting transferred from anther to stigma of different flowers but of different plants. Here it was same, here it was different. Now if you see the statement, now here comes the statement which is very, very important. What is the statement? The statement says that genetically, let me mark this one, genetically autogamy and geitonogamy are similar. Very important thing. Genetically autogamy and geitonogamy are similar. Genetically autogamy and geitonogamy are similar. How this autogamy and geitonogamy are similar? How it is becoming similar? Very simple. If you see the transfer of pollen grain in case of autogamy and if you see the transfer of pollen grain in case of geitonogamy, both the transfer is occurring within the same plant. Here the transfer of pollen grain is occurring inside the same flower of the same plant, right? Here the transfer of pollen grain is occurring between two different flowers but of the same plant. So genetically there won't be any kind of change between these two kind of pollinations. Such kind of pollinations are called what? They are called self-pollination. Why they are called self-pollination? Because it is pollinating its own gynoecium. That is why it is called self-pollination. And these xenogamy are called cross-pollination. Why they are called cross-pollination? Because they are getting transferred from one plant to another plant. Clear? Next topic of discussion is what? Next topic of discussion is agents of pollination. Now this transfer of pollen grains, when a pollen grain is transferred from point A to point B, then obviously it needs some kind of agent to get transferred from point A to point B. Like if you want to go from your house to your friend's house, you need a kind of transport. You need to move from one place to another, right? Similarly here also, if a pollen grain wants to move from anther of a flower to stigma of another flower, then it should have what? It should have certain kind of agents which it will help to move from one place to another. Clear? So these are called what? These are called agents of pollination. The one who helps in the transfer of pollen grains are called agents of pollination. Very simple thing. So I'll keep it simple as possible. There are two different kinds of agents. One is abiotic, the second one is biotic. Abiotic agents are wind and water. 90% of pollination, abiotic pollination, are being done by wind, whereas 10% of abiotic pollination is being done by water. Now the plants which shows wind mediated pollination, it should have a different kind of adaptations. So what are the adaptations? First of all, stamens should be exposed. That means the male androecium should be exposed so that the wind can take away the pollen grains with it. The stigma should be sticky. The pollen grains which is flowing through the air, they should get stuck with the stigma only if the stigma is sticky. The pollen grains should be light in weight so that they can move a long distance in air, a long distance in wind. Numerous inflorescences, that means huge number of flowers are found at a single bunch. A bunch of flowers are formed at a single place of the plant. Numerous inflorescences. Whereas in case of the water pollinated plants, they have a different kind of adaptation. What are the adaptation? The pollen grains can swim due to a whip-like structure. Since they should be traveling inside the water, so they should have certain kind of structure which will help them to swim inside the water. So these pollen grains can swim inside the water due to the presence of a whip-like structure. Okay, and it is protected by a covering which is made up of mucus. That is why it is called mucilaginous covering. Right, so it contains a covering which protects it from the water, because inside the water, if it stays for a long period of time, it might get damaged. And second thing is what? They have a whip-like structure so that they can move from one place to another. The next category of agents are what? They are called biotic agents. They are called what? Biotic agents or living agents. Clear? So what happens in case of the living agents, or what is the adaptations that are required for the plant in order to perform this biotic agent mediated pollination? First of all, if remember always that if you are going to do some kind of work, like if you are going to do some kind of work for someone, right, then that person should pay something to you, right? Very simple thing. If I am working for any person A or person B, then that person A or person B should pay me something. Then only I'll be working. Similarly, since the insects are helping the flowers to transfer their pollen grains from one anther two stigma of another different flower. So that is why these flower gives what? Floral rewards. And these floral rewards are the things also called attractants, the things by which the insects get attracted towards a flower. Why do you get attracted towards a rose plant? Because it smells good, it looks good. Why does a bee gets attracted towards a flower? Because it gets nectar out from there. So these are nothing but these are the floral rewards that the flower gives in order to make sure that the insects can come close to them. They can take the pollen grains and they can transfer the pollen grains from one place to another. These pollen grains have the ability to stuck onto the animal. Like whenever a bee is flying and coming and sitting on a flower, then as soon as it sits on the flower, as soon as it starts taking the nectar, the pollen grains get stuck on the hairs that are present on the legs of this honeybee. Now this honeybee hops onto another flower and there the pollen grains are deposited. Moreover, they have symbiosis with different insects. That means, please, like, it is a mutual understanding between the insect and the flower. Sometimes you will see, you will cut open a fruit or cut open a vegetable, and inside of that vegetable you will see insects. How is that possible that the insects can go inside the fruit or inside the vegetable despite there is no hole on outside part? It is because of this symbiosis. When the pollination process is occurring, then the flower gives a place for those insects to lay their eggs. So the eggs are being laid and the eggs are in symbiosis with that of the small ovary that is present there. So as the ovary matures, the eggs stays inside the fruit or stays inside the vegetable, and with further go they get matured. Clear? So this is called symbiosis. That means I am helping you with this pollination process. You help me by laying my eggs inside you and protecting my own babies. Clear? So this is what is the process of pollination, and this is how the agents of pollination works. Next up, what we are going to see is outbreeding devices. Let us see ahead. So what we have next, we have this outbreeding device. Now remember that the two different types of pollinations are possible. One kind of pollination was self-pollination and the second kind of pollination is cross-pollination. I have already shown you before on that autogamy, geitonogamy and xenogamy. I have also said that autogamy and geitonogamy are self-pollination, whereas the xenogamy is cross-pollination. So a plant always develop certain kind of method in order to avoid this process of self-pollination. Now you might be thinking, sir, why it is necessary for the plants to avoid this self-pollination? Very simple, very simple. Self-pollination, if self-pollination is occurring inside a plant for a long period of time, then there might be some kind of problems like extinction of the plant. Whenever we'll study about genetics, whenever we'll study about evolution, we'll see that this process of cross-pollination or this making of fusing of two different communities with each other helps that particular race to continue the life on the earth. So this process of self-pollination resists the plant to increase their race or to follow their race on the earth. So that is why the plant always tries to make what? The plant always tries to develop method which will help the process of cross-pollination, not self-pollination, cross-pollination. So how this process of cross-pollination is being done? Autogamy, you remember what is autogamy? Transfer of pollen grain from anther to stigma of the same flower, right? So autogamy is prevented by following things. Number one, pollen grain release and stigma receptivity are not coordinated. Very important thing, very important thing. That means the time when the pollen grain is releasing and the time when the stigma will receive that pollen grain is totally non-coordinated. Sometimes what happens, the pollen grains are released but the stigma is not yet receptive. That means the stigma is not yet receiving any pollen grain. So obviously what will happen, the pollen grain will the pollen grain will go away to some other stigma which is receptive at that point of time of some other plant, right? So they are not coordinated. Sometimes the stigma receptivity is there but the pollen grain is not releasing. That means the stigma will receive pollen grain from other plant, right? The position of the stigma and the anther is different. So plants are producing flowers and those flowers are producing the stigma and anther, and they are differentiating the position of these stigma and flowers such like that these stigma and anther is not close to each other to make sure autogamy is not occurring. And the third one and the most simplest one is to produce unisexual flowers. That means a flower which contains only androecium, a flower which contains only gynoecium. Clear? So this is how a plant resists the process of self-pollination and promotes the process of cross-pollination. Same, two different questions, but same answer. Right? Next, pollen-pistil interaction. Very, very important thing, very, very conceptual thing, and a very core part of this whole process of pollination. Now, till now what we have studied, let us rewind once again. We have studied the male reproductive system of plant, the female reproductive system of the plant, the male gamete, the female gamete, transfer of the male gamete and female gamete. We have already completed all these things. Now, what happens when a pollen grain falls on the stigma? What happens? That is our major concern. What is the question? The question is, what happens when this pollen grain falls on the stigma? What is happening? Pollination occurs only when compatible pollen grains falls on the stigma. So this whole process of pollination is successful only if the pollen grain or the correct pollen grain or the compatible pollen grain is falling on the stigma. Like I have a rose flower. Now try to imagine the rose stigma, and on that rose stigma or on that rose stigma, a mango pollen grain has fallen. So is it compatible for the rose plant? No. So the process of pollination won't occur. So the process of pollination will only occur if the pollen grain is compatible and it falls on the stigma. If a pollen grain of a mango tree or a mango flower, sorry, not a mango tree, mango flower, if a pollen that is coming from a mango flower falls on the stigma of another mango flower, then only the pollen grain will undergo pollination. So the pistil has this ability to recognize the correct pollen grain, right? So this pistil has a nice ability to recognize that this pollen grain is correct, this pollen grain is compatible. Wrong pollen grain and pistil inhibits the process of pollination. Now how it identifies, how it recognizes that it is a correct pollen grain? Very simple. It is mediated by a series of chemical reactions by pollen grain and pistil. Very, very important thing. So these pollen grain and pistil undergo a series of chemical reaction, and on this series of chemical reaction make sure that the pollen grain is compatible and the process of pollination should be forwarded. The pollen tube reaches the ovary and enters the ovule through micropyle. Very simple thing, but we'll discuss about this thing when we'll discuss about the double fertilization process. Not now. So make sure whenever you see this point number 5, make sure to point a star. Whenever you are writing down these notes in your copies, make sure to point a star beside it. Why? Because sir has said that we'll discuss about this thing when the double fertilization process will be seen. So very, very important thing. Pollen tube reaches the ovary and enters the ovule through micropyle. But for simplicity, let me explain you here also. So the pollen grain falls on the stigma. Then that pollen grain makes a tube-like structure, and that tube-like structure enters inside the ovule through the micropyle. Now you might be thinking, sir, what is micropyle? I've said what is micropyle. If you don't know, if you don't remember, go to the previous lectures and you will get it out there. Okay, so that is the main point. What I said, that all the lectures are linked to each other. So that is why it is very, very important to see all the lectures, and that is why I'm saying it again and again. So it makes a tube-like structure. Through that tube-like structure, the gametes are coming inside. And that gamete is entering inside the ovule through which end? Through the micropylar end. In the last lecture, and in the lecture number 2 or in part number 2 of this sexual reproduction in flowering plant, I have given a lot of time to explain what is micropylar end, what is chalazal end. So it is very, very recommended to go and view that video and then come to this one in order to have a better understanding. Clear? The last topic of this lecture is what it is: artificial hybridization. Sometimes we as human beings, we have a nice curious mind. Our mind is very, very curious. We are always thinking about something. We always try new things. Sometimes we do this hybridization on our own. What is hybridization? That means the transfer of pollen grain from one plant to another plant. That means from one flower of a plant to another flower of a plant on our own. So whenever we are doing this process artificially on our own, we are collecting the pollen grain from one flower and then depositing those pollen grain on another flower. Then when we are doing all these things, that is called artificial hybridization. Now why this artificial hybridization is done? It is basically done to improve the type and quality of the crop. We need what? We need a better quality of crop, and we need a better type of crop. Nowadays it is quite non-familiar, but if you go to the market to buy rice, then you will see there are different qualities of rice. How all those different qualities of rice were made? It is made by the process of this artificial hybridization and with the help of genetics. We'll discuss about this genetics later on in a very much broader way, but artificial hybridization is one of the most key process for this genetics process. So artificial hybridization is done to improve the quality and the type of the crop. How it is achieved? It is achieved by two different processes. One is emasculation and the second one is bagging. What is emasculation? Emasculation is the removal of anther. The anther is removed from bisexual flowers and they are made unisexual. What are bisexual flowers? Bisexual flowers means they contain androecium, gynoecium both. Now if I remove the androecium from the flower, then the flower contains what? Only the gynoecium. Such kind of process is called what? It is called emasculation. And what is this bagging? Whenever we remove the androecium, obviously the gynoecium is going to get receptive after some time. That means the gynoecium is going to receive the pollen grain from external environment after some time. In order to prevent any other unwanted pollen grain to come and attach to the stigma, we cover the flowers with a small bag-like structure, and that is called bagging. That is called what? That is called bagging. It is called what? It is called bagging. So bagging is basically a process to protect that emasculated flower. Clear? So this is where we'll end our today's lecture. In the next lecture, we'll be discussing about the process of fertilization, how this fertilization is occurring. Remember, I have introduced a term double fertilization while discussing about this point number 5. So we'll be discussing about this double fertilization in the next class, in the next lecture, because it is kind of big one and we want to make sure that each and everything is clear to all of us, right? So that is what we are doing here. So thank you all of you. See you in the next class and in the next lecture. Till then, read these things, obviously get these notes and read your book properly, and stay safe at your homes. So thank you. See you in the next one.